Hi, good morning, good afternoon, and good evening to everyone joining us from around the world. My name is Sabrina Welsh, and I'm the Director of Programs and Operations at the Human Vaccines Project, and I'll be your moderator for this webinar. Today, instead of the news update, I wanted to let you guys know that today will be my last lab meeting with you all. I've accepted a new position, and I'll be leaving HVP in a couple of weeks, and I just wanted to say it's been an absolute honor and a pleasure leading these discussions and meeting with you all. I wanted to thank you for attending, and more importantly, thank you for participating and bringing your insight into these meetings. I also wanted to thank our amazing speakers for sharing their knowledge and time with us. I know your schedules are packed, so I really appreciate your taking the time to join me in these great discussions. If you'd like to keep in touch on my LinkedIn, you can shoot me an email, and just thank you again for joining me. I've learned so much and it's been amazing, so thank you. Happily, I can report that the lab meetings will continue with a new host who I know will do a fantastic job. I'd like to introduce you to Kristen Aboud, who will be taking over the series. Kristen is an exceptional science journalist and has helped guide our highly informative COVID report. And I know that many of you receive it week to week, so please welcome Kristen. Good morning, Kristen. You're on mute. First Zoom test of the day.
Yeah, and a fail at that. But thank you so much, Sabrina, for the kind introduction and also for your amazing work on the global COVID lab meeting this past year. We really will miss you, but thank you so much for all you've done. And I look forward to engaging with all of you and the excellent speakers we have lined up in the coming weeks.
Thank you so much, Kristen. I appreciate it. You're going to do great. And it's a really fun series. I know you'll enjoy it. Just a note before we begin, the information presented today is pre-published data that is currently under peer review. So at the request of the author, we ask that you not reproduce or disseminate the data presented today in any way. Today's speaker is Dr. Peter Brodin from the Karolinska Institute. Dr. Brodin is a consultant in pediatric immunology, combining clinical work at the Karolinska University Hospital with leading a research team of 15 members at the Science for Life laboratory at the Karolinska Institute. The team develops and applies system immunology approaches to study human immune system variation in health and disease and immune development early in life. The work of the group involves the elucidation of heritable and non-heritable factors that shape human immunity, immune system development and adaptation early in life, as well as a research program in clinical application of systems immunology. To enable these activities, the group develops novel experimental and computational methods to more comprehensively and accurately monitor immune systems in humans. Dr. Brodin is also the director and founder of the SciLife Lab, National Facility for Mass Cytometry, and head of the unit for clinical pediatrics at the Department of Women's and Children's Health at the Karolinska Institute, and an elected member of the Young Academy of Sweden and an EMBO Young Investigator. In today's lab meeting, Dr. Brodin will talk about a systems-level blood immunomonitoring study of SARS-CoV-2 patients. During the presentation, please send me your questions using the Q&A function in Zoom. I'll ask our speaker a broad selection of your questions after he's finished the presentation. We'll have about 20 minutes for discussion, and I hope you'll take the opportunity to ask your questions about the data presented today. It is now my pleasure to introduce Dr. Peter Brodin from the Karolinska Institute. Dr. Brodin, good morning. Good afternoon, I should say.
Afternoon, yeah. How are you? Thanks for having me. And
thank you for joining.
It's really terrific. I'll start sharing my screen.
Great.
You guys all see this well. This is not my first slide. Sorry about that. So yes, thank you again. And it's a pleasure to have so many people tune in and listen to what we do. As you accurately said, I'm a pediatrician and a pediatric immunologist, and I combine my clinical work with research at the Science for Life laboratory, which is kind of a technology hub within serving all researchers in Sweden. And so me and my team members have been interested for a while in the development of better methods to profile immune systems in humans. We think we can do this now that technologies have advanced. And this is the sort of framework that we and many others term systems immunology. And what I like to sort of start out by saying when I give these kinds of talks is to kind of define what we mean by systems immunology, because it's become one of those words that people throw around. But what we actually think about is moving a focus from individual immune cell populations or individual proteins and so on to studies which are highly complementary to the sort of reductionist analysis of the past, but that are focused around cell-cell interactions and communication and sort of dependencies. And when I talk about systems level, I don't really mean the entire organism, because that's really not feasible to study. But within a context such as the blood, we can study how cells interact and talk to one another. And that's what I mean when I talk about systems immunology in this case. And so this is what we do, and all of the studies that we perform have this sort of thinking in mind, so to speak. And this sort of work is really made possible thanks to a lot of different technologies that have evolved quickly in the last decade. We're obviously now able to measure hundreds, if not thousands, of features of individual cells, whether it's mRNAs or open chromatin regions, gene bodies, or proteins. We can also measure thousands of proteins in very small amounts of plasma or blood. We can obviously measure the entire genome sequence, DNA sequence of an individual. We could study receptors of variable immune receptors, such as T cells and B cell receptors, and so on. So all of these different technologies give us different perspectives on these different components that make up the human immune system. Another sort of core aspect of everything we do is to monitor something changing over time, because we have to remember that human immune systems are incredibly diverse on the basis of environmental exposures that an individual has encountered, as well as the genetic variation, which is enormous among humans. But by following an individual patient over time, we are able to actually infer relationships between different immune cells and proteins and events that occur that give us added information beyond just individual snapshots. And I think this is another sort of hallmark of everything that we do in our lab. This also has an added benefit of being able to see a lot of different things without the need for thousands of patients, which are usually not possible, cost effective to acquire, to obtain, and so on. Finally, given the fact that the variation among humans is so enormous just due to biological variation in genetics and environment, we have to do everything we can to minimize the other sources of variation in our measurements, such as technical variation. And this has been another thing that me and my team members have worked quite a bit around in the last few years, to try to do everything we can to minimize the sources of technical variation that we introduce as a consequence of processing samples or running experiments. And so we've worked on several areas here. We've developed methods that allow us to freeze whole blood, preserving cell integrity such that single cell analysis can be performed. But this is done directly at blood draw, which minimizes the source of variation that is introduced by cells sitting around waiting to be analyzed and so on. Others, such as the Dara Duffy and Luis Quintana Mercy at the Milieu Intérieur Consortium in Paris, they have developed or worked out a lot of stimulation methods, which also are done directly at the time of blood draw, and which really provides a powerful way to query the functional responses of immune cells directly at blood draw with minimal technical variation, and sort of, in that case, maximizing the biological source of variation that we can measure. So I'm showing here, these are t-SNE plots from mass cytometry data. And here is one of these preserved whole blood samples, preserved directly at blood draw. And we compare that to a PBMC sample from the same blood draw. And obviously you can see that the neutrophils are lost in PBMC preparations, but there are also a lot of variation in, when doing this multiple times, there is technical sources of variation that is always greater in the PBMC fraction than in the whole blood fraction when measuring cell abundances and so on. And this might seem like a sort of minor detail, but it actually has a pretty profound impact on the ability to see changes over time in a patient, or to compare patients that, let's say, respond to a therapy or not, and so on. And the same source of variation is higher when doing stimulation experiments in PBMCs versus whole blood samples, as shown by Dara Duffy and in the group in Paris. All right, so I'm going to talk to you a little bit about some of the work we've been doing during this pandemic. We have had all these technologies, and we've sort of worked out ways that we think help us profile immune systems in humans before the SARS-CoV-2 virus sort of hit. But once we saw that patients were getting sick and you know, patients were having very different trajectories of disease, we decided to try to study the immunology of sort of the determinants of mild versus severe COVID-19. And I'm going to talk about that study. And then as a pediatrician, me and my colleagues around Europe started seeing early in 2020 a new type of patients coming in pretty large numbers, much larger than we've seen before. And these turned out to be patients with this hyperinflammatory syndrome called MIS-C, which is associated with SARS-CoV-2. And so we also began working on those patients to try to understand some of the immunology of that condition. And I'm going to talk to you a little bit about one of those papers we've published. And then currently we're working on understanding the determinants of long COVID, which is patients that suffer from symptoms that persist more than 12 weeks. I won't have time to go through that today, but this is ongoing work in our lab. And so we are really proud to be members of this massive international consortium called the COVID Human Genetic Effort, a consortium that was sort of founded by Jean-Laurent Casanovas at the Rockefeller and Necker in Paris, as well as Helen Su at the NIH and several other colleagues. And we coordinate the Swedish branch of this consortium, and we're running a sequencing hub here at the Science for Life laboratory. And the idea behind this project was that now that this virus became pandemic, Jean-Laurent and Helen and others realized that as so many people are infected with a novel pathogen, and individuals with rare presentations, such as young, previously healthy individuals, would suffer severe disease that stood out from the sort of general population. And those might be identified as having previously unrecognized inborn errors of immunity. And conversely, patients that you would expect to have very high risk, such as the elderly or frail individuals, might get infected or be resistant to infection, might be infected and have a mild condition or be resistant. And those individuals would also be interesting to try to identify now that this virus, which is new to everyone, spreads across the planet. And so we were really excited to get involved in this consortium, and we've been having weekly meetings since beginning of April 2020. And we've learned a lot, I must say. And here are two papers that have come out of this consortium. And I'll briefly talk a little bit about what these showed. So these papers basically showed that two different mechanisms for that could explain a fraction up to 14% of the severe COVID-19 in previously healthy individuals under 50 years of age. And so about 3% of those patients had inborn errors of immunity that was previously unrecognized, either in the viral sensing mechanisms, such as the TLRs, or the signaling pathways downstream, or a larger group of individuals, actually, to me at least, surprisingly large, about 10% of patients with severe disease, had neutralizing high titers autoantibodies, so antibodies that bind the patient's own interferon alpha molecules and preventing them from limiting the viral replication and viral spread. So interestingly, these antiviral sort of autoantibodies to cytokines obviously have been reported in various conditions in the past, but was surprisingly common in these patients with severe COVID-19. And these studies also point to the importance of type 1 interferon and type 1 interferon signaling in preventing severe COVID-19. So I borrowed this figure from the brilliant Akiko Iwasaki, who posted this in October 2020, basically summarizing some of the knowledge that we've acquired to date, and this still holds true, about what determines mild versus severe COVID-19. And what we know is that in cases with milder disease, there is an initial very strong, robust type 1 interferon response. And this is what we think is explaining, for example, the mild disease in children and also in most young adults, which leads to a transient increase in viral load, but then rapidly declining, patients recovering quite quickly from the disease. This is the most sort of common scenario in young people. However, in the elderly, it's more likely to have a delayed interferon response for several reasons. One being that we know that elderly people have a weaker or delayed interferon response, sort of as a consequence of aging, but also because of the virus, that the virus directly inhibits the interferon response, and that is sort of delaying it in the elderly, leading to a higher viral load, but also a more sort of as the immune system tries to catch up with the virus, this leads to a stronger inflammatory response and a more severe disease. Then we have the patients with interferon deficiencies, which are obviously having almost no interferon response, either due to autoantibodies or genetic defects in the interferon pathway. These have uncontrolled viral replication, and these patients are always very, very ill. And have life-threatening disease typically. And so to what can be done about this? Well, there are trials ongoing, and some have shown beneficial early responses by giving interferon to patients at the time of positive testing and so on, suggesting that you can actually try to limit the disease severity in this way, potentially. So I already mentioned this, but the virus has several mechanisms to interfere with the interferon pathway, which obviously explains, sort of underscores the importance of type 1 interferons in this infection, in limiting the infection. But it's important to note that in the cases where the interferon response is inhibited in the cell, the other pathways leading to inflammasome activation, secretion of active pro-inflammatory IL-1 beta and IL-18, they still function. So these patients will then have what we believe to be an unregulated cytokine storm. Some people mention other, but definitely an uncontrolled hyperinflammatory response. And which is not necessarily productive in limiting the virus due to this interferon inhibition. So I think this is one sort of central lesson learned in COVID-19 so far. So me and my team members we teamed up with colleagues in the intensive care unit in Helsinki, in neighboring Finland, and we wanted to study early on using our systems level analysis methods, blood samples from patients with mild versus severe COVID-19. And we collected a number of these patients, and as you can see, some of them were sampled up to 14 times, so very frequent sampling during the severe disease. And I think this is one of the powers of this analysis. This is not the largest population of populations of patients studied, but the very frequent sampling, I think, is a strength of this study. We then wanted to measure plasma proteins, cell populations by mass cytometry. The plasma proteins are measured by O-link assays, which is an antibody-based assay. And the aim here was to separate the immune sort of features between mild versus severe disease. And we found some pretty interesting things, I think. If we focus on some of the snapshots from this is published data, so there's more details in the paper. But if we focus on, for example, the eosinophil population in the blood, This is a population of cells that we typically don't think of as sort of key players in the antiviral immune response. But what we see here using a PAGA type analysis, which is a single cell based analysis with a graph-based abstraction of the data into these two-dimensional plots, where red here indicates dense regions where cells are sort of focused at in the sample. Most of the cells are in that region. And I'm showing you only the eosinophils from one patient, day 0, 1, 2, 4, 6, 8, and 10, and so on. What you can see is that there are changes going on over time. And there's an interesting event that happens a few days after admission to the hospital in this patient. And that is a upregulation of these green cells here, the cells that are eosinophils that express the CD62 ligand, which is a homing marker of cells in the blood. That is a transient event, 2 to 4 days after admission, and then it sort of clears. But what's interesting is that it coincides with the development of acute respiratory distress syndrome in this patient. And it's been shown by others in mouse models and so on that eosinophils that express CD62 ligand likely home to the lung. They respond to gamma interferon, which also seems to kind of, it is high in the beginning, it decreases, but then it kind of levels off right around this, after this event. And so what we think is happening here, obviously this is purely sort of descriptive correlative data following a patient over time. We think that this might actually indicate that eosinophils play an important role in the development of ARDS by going to the lung, being recruited there by gamma interferon and other chemokines, probably. Similarly, there is a population of monocytes in the same patient slightly after this, around day 6, that increases in the blood. And what's interesting about these cells is that they express the fractalkine receptor, CX3CR1. Why is this interesting? Well, it's interesting because this receptor is known to draw myeloid cells out of the bloodstream. It's also known that this receptor usually indicates cells that patrol the endothelium, the blood vessels. And so what we believe here is that these are cells that are activated transiently during the infection or the immune response to the infection. And potentially they're also involved in the pathology of the lung because we know that they can leave the bloodstream using this receptor. So here we have the schematic view of the lung. We know that inflammation in the lung, in the alveoli, during severe COVID and COVID pneumonia involves many different mediators. And a number of studies have been done and some have concluded showing some benefits of IL-6 inhibition, no real effect of hydroxychloroquine, interferon providing interferon seems to have some benefit. But either way, when there's so many different signals going on in the lung, it's difficult to envision one particular targeted therapy that would be able to silence the sort of inflammatory cascades that are ongoing there. And so we've been testing another method to try to mitigate the severe pulmonary manifestations of COVID in ARDS development by trying to limit the trafficking of particularly white blood cells from the bloodstream out into the inflamed lung. And we've done that together with a biotech company here in Sweden called Cancera, which have a specific inhibitor of the fractalkine receptor. And so the idea is that fractalkine might be drawing these cells into the inflamed lung. And if we can prevent them from going into the lung, we might have a better effect than trying to limit the inflammatory signaling that are happening once they're there. The study is still enrolling patients, and we hope to have some conclusive data in a few weeks or months. In this study, we also looked at how cells sort of relate to one another, how they correlate with one another. But maybe more interestingly, we also looked at how this relates to antibody responses. And obviously, we know that patients seroconvert after having COVID, but the levels of antibodies that arise are very variable among patients. And obviously the titers determine the duration of the antibody response to a large extent. And so it's interesting to understand why some patients develop very high titers, why others develop very low titers of antibodies. And so we looked at that using a mixed effects modeling approach. So taking cell composition into account. And what we found was that high gamma interferon and IL-6, which are sort of indicative of a more hyperinflammatory response during COVID, is inversely correlated with strong antibody responses. While some of these other mechanisms, for example, high abundance of basophils seems to be beneficial in the response to by B cells to produce antibodies. That's been shown in other infections previously that basophils can potentiate B cell responses, but I think that might be interesting also in COVID. Finally, we looked at how the immune system responds or changes during the course of recovery. We wanted to understand more whether a patient, if we could identify markers that identify patients as they go from the acute hyperinflammatory COVID-19 response phase and into a more sort of silent recovery phase. And what we're showing here is a multi-omics factor analysis, which is a way to integrate plasma proteins and immune cell measurements into one latent space. And we identify these latent vectors that explain sort of changes during recovery. So here are patients that have recovered, so convalescent patients. Here are patients longitudinally sampled. And as you can see, these patients move over time towards this recovered state. And so we believe that this latent factor 2 increases with recovery. And what does that mean? Well, we see that there are a number of different cells and proteins that change along these axes. For example, obviously there's a decrease in IL-6. That's not surprising because IL-6 is a marker of the hyperinflammatory immune response. But there are other markers as well that might be more sort of novel and indicative of recovery. And for example, we see that there's an increased level of eosinophils, gamma delta T cells, and some NK cell populations during recovery that might be good to monitor to be able to distinguish patients that are on the track of recovery versus patients that have, let's say, a secondary infection, bacterial infection, and so on arising. All right, so the second sort of theme that I wanted to get into is this issue of hyperinflammatory syndromes in children. And these were the kinds of cases that we started seeing here in March and April of 2020. These are kids that come in and they have a presentation which overlap quite well with what we know as Kawasaki disease, which is a condition that every pediatrician is very familiar with, and which basically comes in waves every year, usually after a viral infection, we think. And these patients present as sort of miserable children that are very sort of upset, unhappy, red eyes, swollen red tongue, lymphadenopathy, rash on their hands, feet, maybe scaling, shedding skin on their hands and feet, and so on. They can also develop coronary artery aneurysm, and that's what we are worried about. We treat them with immunomodulatory therapies, and they usually recover quite quickly. However, these patients that were coming in during the spring of 2020, they were greater in number than what we usually see. They were also older, and they were more sick on average than what we typically see with Kawasaki disease. Many of these patients presented with what seemed to be a septic shock, usually due to a failure of the heart to pump strongly enough, and so their blood pressures were low. They were clearly hyperinflammatory with very high values of CRP and high fevers for more than 4 or 5 days. Many were encephalopathic, confused. They have a lot of stomach problems such as stomach ache, vomiting, diarrhea, and so on. And so they were very sick patients, and we were confused as to what this was. Is it a form of Kawasaki disease? Is it a different condition? What we learned quite quickly is that there's a clear relationship between SARS-CoV-2 transmission and this condition. This is numbers from New York during the spring of 2020, with a strong sort of high peak in April, followed by a peak of MIS-C in May. And these are the organs involved, as I already mentioned. And this is the most recent update from the CDC showing the American numbers, where there's been over 3,000 cases, 36 deaths, and an overrepresentation of kids with Hispanic, Latino, or Black ethnicities. And this has been reproduced also in France and the UK. And it's still very puzzling why this is, whether it's due to high levels of transmission in specific communities or whether it's actually a sort of predisposition. We don't know to date. It's also been reported in adults, and so we know now that there is a condition called MIS-A, which is the same condition but in young adults. Yeah, some of these patients obviously very, very ill. So what's going on immunologically? So we decided to take a look at this using the patients that we saw together with, this was done in a collaboration with colleagues in Rome. So we included 13 patients here in Stockholm as well as in Rome. We compared them to patients that we were already studying with classical Kawasaki disease. And these were patients that were sampled in 2017 and 18. So these were definitely not triggered by SARS-CoV-2. We also had healthy children as well as children with a SARS-CoV-2 infection that was mild and did not lead to MIS-C development. So we started out by comparing simply the hyperinflammatory sort of cytokine profiles using Olink assays in adult patients, actually the same patients that I showed you previously in the previous study. And we compared them to the Kawasaki disease patients as well as MIS-C. And as you can see, using this principal component analysis, these are 3 discrete groups. There are 3 patients here that are classified as MIS-C, but have a cytokine profile that is overlapping with Kawasaki disease. And so you wonder if there's a misdiagnosis going on here, if these are separate entities or maybe triggered by another virus and so on. Clearly, there's a discrete, different hyperinflammatory response in patients with acute COVID compared to MIS-C and Kawasaki disease. And the specific mediators, obviously there are hundreds of them, so we don't have to go through all the details. This is work that has been published, but what we see is that Kawasaki disease here in purple, here I have removed the adult patients and focusing in on the children. We see that Kawasaki disease is an overrepresentation of IL-17 producing cells. So it seems to be a IL-17 driven pathology to some extent, with contributions of obviously IL-6, which is high in almost any case, but higher in Kawasaki than in MIS-C and mild COVID. While there's more sort of markers of endothelial activation and endothelial dysfunction in patients with MIS-C than in Kawasaki. And this is shown here as well. So the way we typically treat these patients is we draw a blood sample at the time of presentation. Let me show you, for example, one of these patients here. Then we typically give them IVIG, so immunoglobulins, steroids, and then in more severe cases, in my center here in Stockholm, we give them IL-1 receptor antagonists to prevent the IL-1 beta-driven hyperinflammatory disease. And these patients typically respond very well, and we taper steroids over the course of 1 month. There are a number of markers of successful treatment, such as the ones shown on the right here, TNF and other markers. And these patients typically respond quite quickly to therapy. But we wanted to also understand a little bit more about what might be causing this hyperinflammatory disease. And given the fact that this disease occurs about 1 month out from the initial SARS-CoV-2 infection, we believe, given the fact that most of these patients are SARS-CoV-2 IgG positive, so they have undergone they have seroconverted against the virus, we reasoned that there is evidence, or there's enough suggestions of a adaptive immune response, or sort of an autoimmune response that could explain this disease development. And this is also similar to what has been previously reported in Kawasaki disease, where IgA antibodies to various autoantigens have been reported. So we did a screen of autoantibodies in patients' sera using a whole human protein arrays, and these contain 9,341 human proteins on a glass slide. We add the patient's serum and then a secondary antibody and look for what binds. And then we look for signal differences across patient groups. And obviously these kinds of assays are quite challenging because you get a lot of positive hits, and it's quite difficult sometimes to know which ones are sort of robust hits and real pathogenic autoantibodies and which ones are just sort of spurious signals and cross-reactive antibodies and so on. So we've done a lot of filtering throughout here. We see that there's an overlap of, there's an increased sort of abundance of targets involved in heart development. There's also a lot of pathways involving endothelium and endothelium activation. But there was one group of proteins that stood out the most as sort of specific to MIS-C over healthy and SARS-CoV-2 infected, as well as Kawasaki. And some of them are shown here, which are a group of proteins called casein kinases. So these are enzymes that are widely distributed in the system. And we find autoantibody signals much elevated in MIS-C versus the other conditions. And what made us more interested in this particular pathway was a recent report from Nevan Krogan's lab at UCSF, which had shown that when cells are infected with SARS-CoV-2, they did phosphoproteomic analysis to show that one of the most upregulated kinases were casein kinase 2. And they showed by using a viral inhibitor, an enzymatic inhibitor, a pharmacological treatment, that they could block viral replication in cells in vitro by inhibiting casein kinases. And in fact, what casein kinases seems to do in infected cells was to create this outreaching spurs from the membrane where the virus sits at the top, potentially providing some important function to the viral spread. We don't know that, but anyway, we thought it was interesting that patients with MIS-C had autoantibodies to the same group of kinases that were upregulated by infection. So whether that's a pathogenic role or not needs to be further studied, obviously. There are multiple other hypotheses about why MIS-C develops. One, which is very elegant, which is sort of gathering more evidence now, is that the SARS-CoV-2 virus and the spike protein has a super antigenic motif that can unspecifically trigger T cells with specific clones, specific receptor genes. So remains to be seen. And with this, I'll thank a fantastic group of people, former members, collaborators in Finland for the first study, here at Karolinska for the autoantibody work, Rome with the MIS-C paper, and then the entire international consortium that I collaborate with intensely. thanks so much. And I guess there might be some questions in the chat. I think I saw some.
Yes, we've got a
flashing
few.
By.
So thank you, Dr. Brodin. This data is really interesting and very timely. And you know, the MIS-C especially, it's so scary. I think so many people looked at that, and we were so surprised last year in the US when we started seeing these clusters of kids. It's really scary. So thank you for your work on this and for presenting today. I'm going to jump in. I think I'm going to try to organize these into some lab questions first, because we're getting a lot of like initial, very lab focused questions. Okay, let's take a look. Is there an increase in IL-5 going in parallel with increase of eosinophils?
Yeah, there seems to be. Good short answer.
Good short answer. And I saw you.
I don't know. I mean, I often get the question whether we should block IL-5, and I don't know, but we do see some IL-5, yeah.
Are there studies like that going on? Is that something that can actually be
done? There might be. There's so many clinical trials. I can't keep track of all of them.
I know.
I'm sure there's.
And I saw you answered in the chat, but what is the mechanism by which COVID-19 is able to inhibit type 1 INF production?
Yeah, the virus itself, the SARS-CoV-2 virus, has several mechanisms by which it inhibits and it works in different levels in the cell. So it can inhibit different steps in the transcription of the genes, the sensing, and yeah, so there are multiple mechanisms. And there's been a couple of beautiful papers describing the specific genes in the virus that does this.
And do all strains of COVID-19 have a similar ability to inhibit type 1 interferon, or is that kind of variable by strain?
I think they're all doing that. The sort of variants that we talk a lot about, they usually have other alterations, and not so much this part. I think this is a clear advantage for the virus to be able to inhibit the interferons. So a virus that would alter its ability would probably have a poor fitness, I would expect.
Okay, interesting. Let's see, one factor of different outcomes in different individuals could be the virus itself, since it can mutate and have quasi-species. Did you look into any of these contributing in addition to the host immune factors?
Yeah, so that's a great point. Absolutely true that the virus will vary, of course. And we see that now with the different variants. That's ongoing work in the consortium. Some people are sort of sequencing viruses and looking in relation to the immune side. We haven't done much of that in my group, but I know that's ongoing.
Okay, great. Now a question about TCR repertoire analysis. Would a super antigen show up in TCR repertoire analysis?
Yeah, they do. And there are two papers looking at that at the moment. There are probably more coming, but there are two papers that have already shown evidence of that. One is from Moshe Arditi's lab at UCLA and others. Also a group at Yale led by Carrie Lucas has data on this. So there is a particular TCR V-beta chain that is enriched in these patients. And so that's why this hypothesis is gaining sort of more recognition.
And can you define super antigen for those who aren't familiar with this term? Sort of a...
Oh, yeah, sorry. So super antigen is an antigen that is able to, in a non-specific manner, cross-link and activate T-cell receptors. So normally the way T-cells are activated is by sensing a peptide and an HLA. And that's the sort of traditional way that T-cells get activated. And that's very specific for a particular T-cell based on its receptor. The super antigens, on the other hand, can activate T-cells more broadly in an unspecific manner by just cross-linking T-cell receptor chains and then leading to activation. And if you think about it, if a T-cell population is activated in an unspecific way, that can also drive autoantibody production. It can lead to tissue damage because some of these T-cells...
More broad, just attacking all the tissues and
Right.
Wow. Okay, thanks. Okay, let's see. Do the MIS-C kids have any identifiable risk factors like obesity, atopic allergies, or anything, any kind of markers that you'd notice?
No. These are previously healthy children. Apart from the ethnic differences or ancestral differences that some have reported, other than that, there's nothing clearly seen. They're all mostly the kids we've seen are very healthy children prior to this.
And you mentioned earlier that more Hispanic, Latino, and Black children are experiencing in the US, and sort of broadly, it seems like
Right.
So are people looking in studies for genetic predispositions
yeah.
or other, there are studies going on right now?
That's a big focus of the consortium that I'm a part of, to try to understand, because as you point out correctly, that's a very heterogeneous group from a genetic point of view, and so it's really hard to envision why would there be a sort of similar overrepresentation in such diverse group of people. So I don't know what that means, and I think it's really difficult to say at this point why that is.
Yeah, so sort of some related questions about the disease severity and sort of what's affecting the outcomes. Do you think helminth co-infection versus vaccine efficacy of COVID-19 is it going to reduce efficacy of the COVID-19 vaccines? We've kind of discussed this before in previous meetings. Co-infections, comorbidities.
Yeah, I mean, helminth infections is not very common in large parts of the world, and then it's very common in
some parts.
I know, but it's very different in different parts of the world. And so I really don't know, to be honest. I think that's an important thing to study. Obviously, you might think it might skew the T cell, sort of polarize the T cell states differently. So why not? But it remains to be shown. I don't really know much about that, to be honest.
And sort of relatedly, do you think areas with environmental pollution, and someone asked again, helminth and co-infections and trained immunity and modulation of COVID-19 severity?
Yeah, I mean, those are very different sort of topics in there. Trained immunity, I'm sure, has an important role, and people are trying BCG vaccination as a way to kind of limit disease severity. I think that's a good idea to try. Yeah, I think, all of those, but which factors do, benefit, that's the hard part, to try to figure that out, right?
And in terms of when we're talking about risk, some adults with congenital heart diseases are at a higher risk regarding COVID. Are kids also at a higher risk of either COVID or MIS-C with congenital heart disease? Is that something that affects children like it does with adults?
Well, to our surprise, to some extent, even children with very severe other underlying conditions mostly do very well with COVID-19, and probably because they have a very efficient sort of first line of defense. The only patients that we see as very, very susceptible in childhood, are patients with inborn errors of type 1 interferon immunity or autoantibodies to type 1 interferons and so on. They are very susceptible. But even patients with underlying inborn errors of immunity, other than these, seem to do quite well. And so I think kids generally do very well. There are, of course, exceptions, but generally, yes.
That point reminds me, we had Eleanor Fish from the University of Toronto presented last year on an interferon treatment study. So would administering interferon treatment, would it not be beneficial to the patients who have that deficiency, that sort of abnormality of the inborn error?
So if you have autoantibodies that neutralize type 1 interferons, that usually involves interferon alpha, which is one subclass with many members. But the interferon beta seems to be okay. So there seems to be very much fewer autoantibodies to that. So you could give beta interferon. You could also give lambda interferon, which is typically what we need in the mucosal infections. And so there are studies ongoing in both of these areas. And so, but you're right, of course, if someone has very high titers of neutralizing antibodies, it's kind of pointless to give the same cytokine that the patients will neutralize.
Sure. I think that's why they were talking about, the physicians, when they receive a patient, depending on where they are in their severity and their progress of disease, it may be beneficial or may be completely useless, depending on when that person is seen. So very interesting. I'm curious to see Eleanor's upcoming data for the study. It'd be good. Okay, back to the lab questions. In your multi-omic analyses, did you see any obvious connections between the immune system and angiotensin pathways potentially mediated through the ACE2 receptor?
Oh, this is a really good point. I mean, the ACE2 receptor is definitely key here. We did not see much in that area at all, but obviously there's been discussions around, expression levels of the ACE2. We know it differs between the young and old and people with hypertension, ACE2 blockers, and so on. But in our case, we did not see so much signals of that. But there might be, we didn't necessarily look into the respiratory epithelium, and whether the ACE2 receptor there is activated and expressed and so on, which is probably the more relevant part.
Okay, gotcha, thanks. Does CCR2 play a role in trafficking of myeloid cells and tissues during COVID?
I think it does, and CCR2 is definitely kind of a sort of a, we see it in many different tissues with tissue damage in the liver and so on, and even in the brain, and so I think it probably does play a role.
Okay, how important is it in your opinion to inhibit viral replication? That seems
well, that
like
is
a
kind
pretty
of key,
obvious
right?
one you want to.
I mean, I think that is, I think this whole interferon story points to how important early in disease, obviously it's in the early phase, that's when you really want to limit viral replication and sort of lower the viral replication as much as you can. I think it is very important, and that's, the interferons have really proven to be the key to determine mild versus severe disease here.
Great. Okay, eosinophil infiltration is suggested as a marker in SARS-1 and in RSV-infected infants, particularly in animal models after challenge. Part of a Th2 skewed immune response. How damaging are the eosinophils that you see in the SARS-2 patients? And you mentioned that earlier. There seems to be correlations.
Yeah, we basically just see that they pop up right before the patients develop ARDS. That's what we've done, basically. We haven't tried to limit or mitigate their function and so on. But I do think all of these pieces, do fit together into sort of a underappreciated role for eosinophils in this pathology. And I do think we should pay more attention to them, even, in viral infections. And as this attendant correctly points out, there is evidence of that in the past. We just don't typically focus so much on them, but I would like us to do more,
Yeah, it'd be interesting to see. Okay, is there a supersite on the spike protein? If there is a supersite on the spike protein, is there concern with using the same spike protein in vaccine regimens? Because I know a lot of them are targeted towards spike.
That's a very, very good point. But what's important is that there's a particular furin cleavage site on the spike that is important for the super antigenic feature. And so I am less concerned about the vaccines. When I initially sort of read about this and started looking into this, I was getting very concerned about vaccines causing MIS-C. But to my knowledge, there is not a single case reported for vaccine-induced MIS-C. And given the fact that the spike proteins are slightly different in the vaccines, and the way that they have, they are stabilized in fusion mode, most of them, except for the AstraZeneca one, right? But still, they all lack this furin cleavage site. So I think I'm less concerned about the vaccines in this case.
Sure. Okay. We've got a couple of questions around BCG. I know we mentioned earlier that the vaccination with BCG might limit or affect disease severity or infection. So could you explain kind of what that mechanism is, and would you agree that BCG is capable of sustaining type 1 interferon responses despite COVID infection? If you could talk a little bit about that as well, that'd be great.
Yeah, I mean, the idea behind this is trained immunity, basically, so that by giving BCG, you would reprogram particularly the myeloid compartment into by epigenetic remodeling, and that would then turn out to be beneficial once COVID or SARS-CoV-2 would infect you. That's the logic, and whether it's capable of sustaining type 1, I think remains to be shown. In theory, this might work, and these trials are ongoing, and to me, they seem to be very well planned and executed. So I think we should get some conclusive data on this topic quite soon.
Okay, relatedly, a question now, what's peculiar about the immune response to the post-fusion configuration of the spike?
Well, good question. I'm not sure. I mean, the only thing I know is that the vaccines typically use the pre-fusion one, because that's what you would want when you want to develop neutralizing antibodies that might be, preventing fusion or preventing infection. Whether there's anything peculiar, I'm not quite sure. There might be someone who knows, but I don't really know if there's,
So going back to our conversation about MIS-C and environmental factors or different predispositions, why are we seeing much lower CFR and almost negligible MIS-C in Southeast Asian countries? So a dark participant note in Thailand is 70 million people, 30,000 reported cases, and no reported cases of MIS-C. So is this something worth studying? And do you know of any studies that are checking out?
No, I think this is a terrific point. And I'm also extremely puzzled by this, especially given the fact that Kawasaki disease, which seems to be a very much related disease, is so much more prevalent in East Asia. And so why this is, I don't know, but it is definitely a puzzling observation that seems to be very robust. And yes, it must be studied. The genetics underpinning this is definitely in the, it's under study, it's going on. And so I really hope we can get some clarity on this.
Yeah, definitely. Okay, so we're almost at the end of time. I just wanted to give you an opportunity. We've had so many interesting data sets, and it sounds like the evolution of these multiple technologies and these large global consortiums have really contributed to increasing our knowledge base, and the collaborations are speeding along, the data and how quickly we're able to access data. Is there, what's coming next for you? And if you could comment on, if you had unlimited resources, or if you could access one resource you don't have currently, what are you looking to do next in your studies?
Yeah, this is really good point. I mean, we would love to grow on the populations that we study, because we know that obviously human immune systems are so incredibly diverse, but there's so much to learn by taking more of the inter-individual variability into account. And so if we had unlimited resources, we would basically scale up these studies to be able to take more of the genetic and environmental variability into account. And I think we would learn so much more. So that, and also move sort of across the globe and study different populations would be absolutely important. So that's what I would do.
Great. Well, best of luck. We're looking forward to seeing what comes next in your research. And if anyone has questions for Dr. Brodin, feel free to reach out and we can connect you if you have additional populations. Yeah.
Thank you so much. This was a pleasure. Great questions.
Thank you so much. We appreciate your time. I'm going to share a couple of slides and then we will wrap up.
Thank you.
Okay, so thank you again to our participants and to Dr. Brodin for presenting today. We really couldn't do it without you. These conversations are so interesting, and having your input and interrogating the data makes it so much better. So thank you for joining. I invite you to join us 2 weeks from today on the next Global COVID Lab meeting, which is scheduled on April 22nd. Our next speaker will be Dr. Nir Barzilay from the Albert Einstein College of Medicine, who will be talking about immunosenescence in COVID-19 vaccines for the elderly. I also wanted to note that the application portal is now open for the Michelson Prize's Next Generation grants. Presented by the Human Vaccines Project and the Michelson Medical Research Foundation, the Michelson Prizes are given to support promising young researchers who are applying disruptive concepts and inventive methodology to advance human immunology, vaccine discovery, and immunotherapy research for major global diseases. If you're age 35 and under, or you know someone who'd be interested in applying, please check out the link below that I'm showing on the screen. And if you're interested in more research on COVID-19, please sign up for the HVP COVID report. In this newsletter, we present insights from experts all around the globe and highlight the latest scientific articles and data. And lastly, please feel free to visit our website and follow us on LinkedIn, where we'll upload a recording of this webinar for you to review. And with that, I'll say thank you again for joining, stay safe, and we hope you'll join us on the next Global COVID Lab meeting. Thank you so much, everybody. Have a great day.